I'm happy to introduce Mariana Borsa, who just started her, maybe not independent career, but her independent lab in Basel. Actually, she's starting in next month. And she's the most famous for her work on asymmetric cell division in T cells. So we'll hear now what she's up to and what are her newest discoveries.
Thank you very much for the invitation. So today I'm gonna share with you still unpublished work. It's hopefully soon in press, but something I have done during my postdoc here in Oxford. And it's about how the inheritance of mitochondria shapes T-cell fate decisions very early on with the help of autophagy. So I know this is like an immunology crowd that is listening to the seminar, but I just would like to tell a bit my motivation for this work. So I'm very interested in stem cells, and as a trained immunologist, I have used T cells as my model to study stemness. And if we think about CD8 T cells specifically, they can be subdivided in, of course, many more subsets than what I'm putting here. But in a basic way, we have cells that never saw the antigen before, that are the naive cells, that can then lead to the rise of memory precursors and effector cells. And you also have long-lived memory cells. And I'm highlighting here the naive and the long-lived memory as the stem-like cells, and these are the cells that I'm very interested in. So if we think about the course of an immune response to an acute viral infection, for example, very like one single cell can give rise to already this memory precursors and effector cells. The effector cells being the ones that populate, that are really like expanding very fast and being majority at the peak of T-cell responses. But then the memory precursors that are already there give rise to the cells that are long-lived, and they're going to be important in the context of the re-challenge. And what I would like to highlight here is that this early fate divergence really relies on early events that occur from the moment of the immune synapse of this naive cell, where if the cell receives the three signals, 1, 2, 3, meaning the one that comes from the MHC peptide, co-stimulation, also like have the right cytokines around, then this naive T cell can undergo its first division, already forming a polarization axis. And this polarization axis is going to be maintained until the end of the mitosis, and it's going to then give rise to daughter cells that have distinct potential fates. And that's what I have been working over the last, I think, more than 10 years now. And it's called asymmetric cell division, which is a conserved mechanism that is observed not just in T cells, but in other stem-like cells. And that's going to be important. That's very important to maintain stemness. And I'm going to tell you how this works, for example, with T cells. So if we think about, let's imagine that the antigen presenting cell was on this side. So this would be what we call the proximal side of this mitosis. There is recruitment of different molecules to that side, including surface markers like cytokine receptors, the TCR itself. But there are also layers, cytoplasmic layers of asymmetry that are formed, such as the accumulation of certain transcription factors on this proximal side, and also like different metabolism. So basically you have the formation of different layers of asymmetry, and this is the reason why the daughter cell that comes from this proximal side has a potential different fate from the cell that's on the opposite side, being the one that is closer to the synapse, more prone to become an effector cell, and the other cell more prone to become a memory cell. It's not black and white, of course, because we know that cells can be in different environments, and the environment is also going to have an influence on their fate, but there is definitely a cell intrinsic bias already formed from the first mitosis. So during my PhD, when I was in Zurich, I worked with more like descriptive, it was more descriptive work most of the time, basically looking at how different T cells can divide asymmetrically or not. And back then we found, at least in murine T cells, that naive and memory cells are able to undergo asymmetric cell division, while cells that are isolated from aged individual animals or cells that come from mice that were chronically infected, these cells cannot undergo ACD. However, we also found a way to modulate this process. So we found a way to boost asymmetric cell division. And this was done by transient mTOR inhibition using rapamycin, but also some other inhibitors that had a similar effect. And then we found that we could also rescue asymmetric cell division in these cells, and that this had a positive effect in the formation of memory responses. And importantly, this was not linked to metabolism, because this transient, it was a transient inhibition that was washed away. So it was really an effect that we had for the time of the mitosis that increased. Increasing these asymmetries, we increased the formation of cells that are memory-like, that are basically the cells here that are clear of lots of cell cargos. So asymmetric cell division is something that drops with age, and we know that with age we also lose stemness, and this is also particularly detrimental to memory responses. And there is another cell biological mechanism that also deteriorates with age, that's called autophagy. Just in a very simple way, autophagy is this recycling pathway, degradation and recycling pathway that we have in our cells, that relies on the formation of this double membrane structure called autophagosome, that is going to rely on lots of autophagy genes that we call ATG genes. And I'm showing this because I'm going to show knockouts of these genes later. And these autophagosomes fuse with lysosomes, and then you have degradation of the cargo that was inside the autophagosomes, and then this can be used also as building blocks by the cell. So both asymmetric cell division and autophagy are mechanisms that are important for memory formation, for stemness, and that deteriorate with age. And that's why I came to Oxford to do my postdoc with Katja, who then soon moved actually to the Max de Bruck Center in Berlin, but I stayed here. And then I came here to basically investigate whether autophagy regulates the rise of these asymmetric fates very early on. So the first thing we did soon after lockdown was kind of over was to send these daughter cells, these first daughter cells, for proteomics, and this was done in the lab of the Rinkentrell in Dundee. And the idea of doing proteomics of these daughter cells, because up to that point we just knew that the cells had different transcriptomes, but as we wanted to know what would be the role of autophagy, and autophagy is something that's going to then be important for degradation of what's already translated. We thought that proteomics could give us a hint of what is autophagy targeting, and whether there are different differential autophagy upon division. So basically we sent this daughter cells, this first daughter cells to proteomics, and then this was done like in different experiments, and the variation was quite big. But when we pulled all together, what we could see is that there were some genes, well, some proteins that were different between these two populations, and that many of these top proteins, differentially inherited proteins, were proteins that were linked to mitochondrial metabolism and biogenesis. So that's the moment when we actually started looking at mitochondria. This was first like an unbiased approach, like to see what we would find, and then we saw that mitochondria would be a potential target. And then importantly, we also sent daughter cells, the first daughter cells from animals, and this is basically, this is basically heat map showing like this mitoproteins that we found that were differentially expressed. And you see that between the cells that I call high and low is because they are high or low for a marker of effector cells. So these are the effector cells and these are the memory-like cells. And you see that the expression is different, and some proteins are actually absent in one or the other cell. So there are differentially expressed, differentially inherited mitochondrial proteins. And when we isolate cells from autophagy deficient mice or from mice that were old, very old, like more than 100 weeks, we couldn't really find these mitochondrial proteins being differentially inherited. And also the number of proteins that was differentially inherited was lower. So this suggested that autophagy contributes to this asymmetric inheritance of proteins involved in mitochondrial function. And then the first thing we did was to see whether there is different amount of mitochondria in one or the other cell. And we tried this in different ways. We did some electron microscopy. We also just like stained for TOM20 as a marker of mitochondria. And we couldn't really see difference in mitochondrial mass between the other cells that are effector-like or memory-like. But then we looked at types of mitochondria. And that's when we found that mitochondria that express superoxide, which can be stained by mitosocs, if these are basically mitosis of wild type and knockout cells, and we could see that in the wild types there was asymmetric inheritance of these mitosocs, and in the knockouts this was not happening. And this can be quantified, and this type of graph is going to pop up later as well. That's why I'm explaining it a bit here. But basically here, using this ratio, P1 is the pole, like the daughter cell that has more CD8 as a surrogate marker for effector-like cells. And the more far from zero, using this ratio, more asymmetric is the distribution of what you're looking at. And we have this threshold of 0.2 based on the distribution of DAPI itself that has a bit of variation. So basically knowing that nucleus is the same on both sides, and that the range of DAPI would be around this gray area. Basically everything that's above or below that we consider asymmetric. So here's this 0.2 threshold means that you have 50% more of a marker on one side than on the other side. And as you can appreciate, in the wild types, you have more mitosocs on the same side as CD8, so on the effector-like cell. And in the knockouts, this distribution is a bit more random, but also more symmetric. So this led us to conclude that asymmetric inheritance of mitosocs is lost in these autophagy deficient cells. The problem is that mitosocs cannot really be used for functional readouts. So of course, like we see that there is asymmetric distribution, but what does it mean? Does it change the fate of the cells? And T cells don't really like having mitosocs around. So basically what we did is to team up with Pekka. Pekka has developed in his lab an animal model where the outer membrane protein 25 of the mitochondria is bound to a SNAP tag. And a SNAP tag is like a modified enzyme. It was originally a DNA repair enzyme, but basically they modify it in a way that doesn't exert its function anymore, but it still binds to a substrate. And the substrates are brilliant because they are cell permeable and fluorescent, which allows us to do sequential tagging of newly formed units of mitochondria based on OMP25 biogenesis. So in T cells, that's the timeline we have used because we are interested in the first mitosis. So we have T cells that are isolated from a mitoSNAP animal, and we activate the cells in presence of tamoxifen, so it's an inducible expression of this tag. And that's why on the next day we come with a SNAP substrate. Here it's a substrate that is in the far red range, for example. And then we wash it out. So every mitochondria that had a OMP25 that was generated in this time frame will be stained with this far red dye. And then we let the cells keep, put them back in culture, they keep doing what they have to do. And then more or less like a day later, we come with a SNAP block. That's a substrate with no color. And then we let the cells also like stay for 2 more hours in culture. And then we stay with a second substrate, which means that here we have older pieces of mitochondria being stained with this dye. And here we have very recent pieces of mitochondria. And we didn't expect what we saw, but what we could conclude from our stainings, and this was very reproducible throughout time, is that first, you have actually a lot of biogenesis of mitochondria very fast. So you always have some young mitochondria being produced even in few hours. And the second thing is that the young, maybe I zoom in if it becomes easier, the young network of mitochondria is actually being built on the old network. So there's most of the mitochondria, the new mitochondria, new pieces of mitochondria are not really isolated from the old mitochondria. So they are at the same place. And then the consequence of that, and I hope I can, oh yeah. The consequence of that is that if you look at the other cells, first other cells, and you look at the SNAP tag staining, basically you have cells that keep both mitochondria, older and younger, and cells that get rid of this old mitochondria. And this, of course, could be because of different reasons. It could be because of segregation through asymmetric apportioning of mitochondria, but also might have a contribution of mitophagy. So we looked at these two things separately. First, looking at mitosis. If you look at the old mitochondria, and here, from now on, most of the time I'm gonna just show old mitochondria as the young and the old were at the same place and were being co-inherited. So looking at old mitochondria here, you see that in the wild type, in this mitosis, for example, this is very asymmetric, this distribution. And in the knockout for autophagy, you have much more mitochondria because mitophagy is impaired. And also this distribution is more symmetric. And this is what is also depicted here in this graph that I explained how to read before. So it's much more asymmetric in the wild type than in the knockout. So there is asymmetric segregation of mitochondria or of types of mitochondria. But then we also wanted to look at mitophagy or autophagy as well, and then to kind of not exclude, but isolate this first mitosis from our analysis. What we did is to sort, so basically doing this very, very similar type of tagging. We sorted the cells that are positive or negative for this tagged mitochondria, and then we put them back in culture. And then obviously the cells that are mitosNAP low remain mitosNAP low. And there are also some mitosNAP low cells in the knockouts. It could be because of lack of biogenesis, it could be because of segregation, we are not sure, but there are not so many, but they are there. But if you get the cells that are mitosNAP high after the first mitosis and you put them back in culture, if they are wild type, in 3 days, they clear up all this mitochondria. And in the case of the knockouts, you still have a large proportion of cells that keeps them. So there is a contribution of autophagy also to the rise of the cells that are devoid of SNAP tag labeled mitochondria. So there is basically a synergy between segregation and degradation to the generation of the cells. So then the next step we wanted to figure out is like, does it matter? Like, then what we did is that we transferred, we isolated the cells that are mitosNAP high or mitosNAP low, and we did adoptive transfer experiments to figure out whether this would have any role in the fate decision of the cells. So these these mice are now with an OT1 background, which means that we can use OVA as like a cognate antigen to activate the cells. So we transfer the cells that are mitosNAP low and mitosNAP hide to new hosts, and we wait for 30 days until we came with the challenge with the Listeria monocytogenes over. And the reason why we waited for 30 days was really to benefit cells that would be able to live, like to survive without antigen around and re-expand upon re-challenge. And then after that, we did a blood kinetics and looked at the cells at memory phase in the spleens. And as you can see here, there is upon re-challenge, there are more mitosnap low cells, so the re-expansion of mitosnap low cells is clear. And if you look at the spleens at the memory phase, this mitosnap low cells outnumber the mitosnap high progenies, both in percentage within CD8 cells, but also if you look at total numbers. So this made us conclude that mitosnap low cells are cells that retain better memory potential, being the mitosnap high cells more short-lived. And then why is this happening? So why do mitosnap low cells are better than mitosnap high cells as memory cells? And then there are several reports in the literature saying that, for example, mitochondrial architecture can play a role in metabolism, of course, and that this can influence T cell fates. And that's why I teamed up then with Ed from the lab of Mike Dusting here in Oxford, we kind of set up a pipeline to image the cells in 3D and then look at mitochondrial architecture in a more unbiased approach, 3D rendering these organelles like individually. And then with this, we can do U-maps of individual mitochondria. These are not cells, these are basically mitochondria inside the cells. And then in the mitosnap low cells, of course, all the mitochondria are low for the SNAP tag. In the mitosnap high cells, you have a mix of types of mitochondria, being some mitochondria also low for the SNAP tag, and some mitochondria intermediate, some mitochondria very high. And what we could see is that the mitochondria that are very high for this SNAP tag, that retain the SNAP tag, so the older mitochondria are the ones that are more complex, which means that they are less round, they are more like elongated, and also and less compact, which would suggest that these mitochondria are more difficult also to undergo autophagy, because autophagy mostly is going to eat, like basically engulf mitochondria that are smaller. So the architecture already suggested that this old mitochondria this mitosnap high mitochondria are indeed older and more difficult to be mitophagy targets. But then we also looked at the metabolism of these cells that inherit or not this SNAP tag labeled mitochondria in different approaches. One of them just doing the common seahorse, where we could see that the mitosnap high cells are more glycolytic, which would fit with the idea that they are effector cells. And we also used another technique that allows you to do assessment of metabolic reliance by flow cytometry. That's called Zenith. I'm going to talk a little bit about this technique in a few slides, but it has been published a few years ago by Rafa Arguello, and it's very nice because it allows you to have single cell resolution of metabolic reliance. And then on the other side, the mitosnap low cells are more quiescent and have higher basal respiration. So overall, like the metabolism of the cells would indeed fit what the literature has reported before for effector memory cells. But the interesting thing here is that we show that this is happening from the first mitosis, and that this is really reliant on the inheritance of this mitochondria that are not undergoing mitophagy and that are becoming older. So we also did it. We also did an experiment by looking at this first division in vivo. This is one of the things that the reviewers asked us to do, to show that this is not like just an in vitro artifact. So what we did is to transfer this OT1 cells naive to new hosts, and then challenged the mice with Neisseria monocytogenes infecto. And then we looked at the cells that divided just once. And then we looked at the cells that divided just once based on the inheritance of all mitochondria. We could see that the cells that are mitoSNP high, for example, are cells that express more granzyme B. So also this in vivo first order cells, generated first order cells, also show different phenotype based on the inheritance of old mitochondria, being the ones that inherited old mitochondria more effector-like. So finally, we until now everything sounded a bit obvious to us, and then we thought, let's just see whether we find something that we cannot think about by doing better characterization of this mitoSNP high and mitoSNP low daughter cells by proteomics, again with the help of Doreen Cantrell's lab. And as you see, it was a bit disappointing. This volcano plot is quite shy. This is again pooled data from different experiments where there is of course variation, especially in proteomics. But one interesting thing we found is that in the mitoSNP high cells, there was a lot of proteins linked to proliferation, so lots of kinesins. And then of course, like proteins such as HIF1 alpha that are linked to mTOR activation, which is also something that you would observe in effector cells. So from the few proteins that we could see that were asymmetrically inherited, it's again like suggested that the mitoSNP high cells are more effector-like in comparison to the mitoSNP low. But then because this didn't give us much more information, we decided to do some transcriptomics of these cells, and this was done with the help of Amir, who was a postdoc in the lab of Katia in Berlin and is now in New York. And what we saw is that overall, like when we look at the signature the transcriptome of this mitoSNP high and mitoSNP low cells, again, there would be higher, like this mitoSNP high cells would resemble effector cells because they have more expression of genes linked to proliferation and cytotoxicity. And the mitoSNP low cells would be more quiescent and would have more higher expression of genes linked to memory signature. And if we look at pseudotime analysis, you see that the cells actually, you have to imagine that they just underwent one division, so they were one cell just a few hours before, but so they are occupying similar space in these UMAPs. But if you would look at the trajectory that they take, the mitoSNP high cells are the ones that are reaching the top of this UMAP here, where you have actually a cluster of cells that is very high, that has very high expression of cytotoxic molecules and pro-inflammatory cytokines, and that are terminally differentiated. So also looking at transcriptomics, it would again point out to the fact that the cells that retain this old mitochondria are the ones that are becoming effector cells. So one interesting thing that popped up in this, I'm just going to look at the time. One interesting thing that popped up when we look at this transcriptomics is that we found a cluster of cells that had a very high expression of genes that were linked to one carbon metabolism. And this is something that, I'm an immunologist and not like a mitochondrial biologist, or even like now I'm learning more about metabolism, but it seems to be something very common that cells that have old or damaged mitochondria have higher, rely more on one carbon metabolism or trigger one carbon metabolism. And this is something that then we looked at, we focus on towards the end of this story. So one carbon metabolism does lots of things, and I probably don't know all the things that it does. But it's basically comprising what we call the folate and the methionine cycle. And it has enzymes, so basically it has enzymatic reactions happening both at the cytoplasm but also in the mitochondria. And we had here in red, I'm highlighting the genes, or actually the proteins that have encoding genes that had higher expression in the mitosnap high cells, the ones that had old mitochondria. And you can, I think, appreciate that you have basically both the cytoplasmic and the mitochondrial versions of these enzymes being highly expressed in the mitosnap high cells. And this, amongst the downstream pathways that the one-carbon metabolism contributes to are like methylation of genes, nucleic acid synthesis, also redox control. It has been suggested in different cell types, including T cells, that increasing one-carbon metabolism is a way to cope with redox stress, which is more pronounced when you have damaged mitochondria around. So what I did is to then look at whether these mitosnap high cells indeed rely on one-carbon metabolism. And the first experiment I did is to use the Senate technique that I'm going to explain a bit more now. So the Senate technique basically is a way to, they have reported that, and this is the reference, they have reported that there is a very strong correlation between translation and ATP production. So we use translation as a measure of metabolic reliance. And translation is something that you can measure with the incorporation of an amino acid analog that's called OPP. And with OPP can, with click reactions, you can make it have different fluorescence. Here I have used the one that's Alexa Fluor 594. But basically what you can already see is that the mitosnap low cells, here is basically the control with no inhibitors around. If I just look at translation rates in the mitosnap low cells, they are indeed much more quiescent than the mitosnap high cells. So already pointing out the fact that the cells are more metabolic active and effector-like. And then if you come with inhibitors for the mitosnap low cells, they are already so quiescent. So this is kind of the baseline that the inhibitors don't do much. So here's basically using an inhibitor of glycolysis, that's 2-deoxyglucose, and of respiration, that's oligomycin. So basically, if you shut down both glycolysis and respiration, this is when the translation rates or ATP production and translation rates should be the lowest. And you see that there is a little drop that's not even statistically significant. So the cells are super quiescent. But then when you look at the mitosnap high cells that are more metabolically active, this is kind of the baseline we can get with 2DG and oligomycin together here. And when we come with the one-carbon metabolism inhibitors, some of them don't do much, but the ones that inhibit this enzyme called SHNT1 or 2, and this one up here, the PHGDH, there is a drop in these translation levels, which then suggests that these cells are indeed doing active one-carbon metabolism, and that when we inhibit these enzymes, there is a drop in their metabolic rates. So, of course, this is like somehow, it is a functional readout, but it's not really, it's a bit indirect. It's not really showing the flux of metabolites in the cell. And that's when we submitted the manuscript, we were asked by reviewers to also look at the metabolome of the cells. So what we had to do is, like, again, to sort this mitosnap high and the mitosnap low cells and send them for metabolomics. And we did fluxomics. So we did like carbon-13 label. We use carbon-13 labeled glucose and also serine. And I'm going to explain to you here now. So, and why glucose and serine? Because you see that glucose is here, up here in the pathway. So before this PHGDH, and serine is a very important amino acid for one-carbon metabolism in T cells. So that's why we did it with both. So if we look at, this is using both here glucose labeled and serine labeled. You can see that in the mitosnap high in comparison to the mitosnap low, especially when we look at serine labeled, you have more labeling of glycine with this 13 carbon 13 isotope in the mitosnap high cells. I think this is very clear here. There's also higher abundance of this metabolite. And when we look at, and now I have to, I apologize because now I have to think of it again. And this is basically in the knockout. And in the knockout, you see that there is not much of a difference. So basically what it means is that a knockout for autophagy, of course. So it means that when you are a wild type cell, your daughter cells will have different metabolism. So one of them will rely more on carbon metabolism than the other one. And this has now been shown also for different metabolites. I just picked glycine because I think it makes it easy to understand that it's coming from serine to glycine, or from glucose somehow to glycine, and that there is a higher flux of this metabolite in the mitosnap high cell. But in the knockout cells, this difference is not so clear. So here, if you compare glucose to glucose in the knockout, there is not much of a difference, and serine to serine in the knockout, there is not such a big difference as in the wild type cells. So another thing that we found is that when we do serine depletion, so when we get rid of this amino acid that's so important for one carbon metabolism, we actually can revert a bit this phenotype in the mitosnap high cells. And that's what I'm showing here. So basically, if we get these mitosnap high cells and we put them back in culture with or without serine, and that's what I would like you to pay attention here. You see that when there is no serine, the cells express less CD44 and CD25, which are activation markers for T cells. And this is what is quantified here. So basically you have, if you look at without serine and glycine depletion, you have in the high, they are a bit more activated, and there is a huge drop basically in both subsets, but there is a huge drop also in the mitosnap high of these activation markers if there is no serine around, so they become more quiescent somehow. And if you look at survival of the cells, here basically it's a condition where you have serine around, and this is when you deplete serine in this second bar here. So you see there is a basically more than doubles the survival rates of the cells. And if you block this, if you block the uptake of this enzyme here, you see that there is not much of a difference, which suggests that it's because you have more, basically that this different metabolism is happening because you have more uptake of serine and not because you're having more conversion of glucose to serine. So this was the control. Maybe now that it became too complicated, but basically the idea here is to say that these mitosnap high cells rely on serine to have activated one carbon metabolism, and that if you deplete serine, then the cells become more quiescent. So this might be a way to manipulate the fate of the cells, modulate their fate. So overall, what I would like to you to have as a take-home message is that when you have this first division of T cells, CD8 T cells, you have one cell that ends up keeping mitochondria for longer. And this is because it's a mix of segregation of this mitochondria upon mitosis, but also because these cells here are doing less mitophagy, and then this mitochondria is staying there longer, while this other cells here are already devoid of these organelles, and this is mostly reliant on very fast autophagy, very fast mitophagy of this newly formed mitochondria. And we don't really understand why these cells that are quiescent are doing mitophagy so actively, but they are definitely trying to keep them themselves free of damaged mitochondria. And then the consequence of it is that you have changes in T cell metabolism in the cells that inherited the damaged mitochondria, and then that they become short-lived cells, while the cells here have more memory potential. So with this, I would like to thank Katja for the mentorship over the last 6 years. I would also like to thank Anna. Anna is now also like an independent researcher. She opened her lab in Barcelona, and she knows a lot about mitochondria. I learned a lot from her. There are members in the Oxford lab that contributed also to this work, and also in the Berlin crew. People from the lab of Mike Dusting here in Oxford, and also from the Clark group. The Ring and Linda for the help with the proteomics, and the lab of Pekka and team for providing the mouse, but also for providing me with insights of how to use this model as they use it in other cell types in their lab. These are the funding bodies that helped this work to happen. And as it has already been announced, I'm an incoming assistant professor now. Very soon, I'm going to start in Basel, and I'm actually looking for PhD students and postdocs. I still have some vacancies, and yeah, if you are interested in this type of work, I would be happy to talk to you. And if I have 1 minute, I just wanted to say one thing, like what is the lab going to do? And I'm gonna just go to the last slide of the presentation, because I have just 2 minutes. But basically, this is the last slide. Basically, this model that we have for mitochondria, we also have now optimized for peroxisomes. Peroxisomes are a very neglected organelle, but very important for lipid metabolism. We don't know much about their contribution to T cell or even to immune cell fates. But I have done already a lot of preliminary work with this peroxisnap model. And the very intriguing thing is that, and still looking at first mitosis, something that I don't want to do anymore. I want to look more at cells that inherit or don't inherit, independent of this first division. But using still this first division model, what we saw is that similar to the mitochondria, cells also have asymmetric inheritance of peroxisomes. But the consequence of inheriting no peroxisome is the opposite of the one inheriting mitochondria. So cells that inherit old mitochondria become short-lived and effector-like, and cells that inherit old peroxisomes are long-lived and memory-like. And that's why I usually put this analogy here, that mitochondria age like milk and peroxisomes seems to age like wine. And this is something that I do not prove yet. It's one of the things I would like to do in the lab, is basically to understand how aging of organelles happens differently, and the consequence of inheriting these old organelles to T cell function in the context of aging. So also, like, is an old person less capable of doing this asymmetric, of having this asymmetric inheritance of these organelles? Does it have a consequence? Do they keep these old organelles for longer? Is it different for peroxisomes, that as it seems that peroxisomes actually, when they're protected from degradation, they become better? So this is what the lab is going to work on. And with this, I would like to thank you for your attention.
Thank you a lot, Mariana. So now we have time for questions. So either raise your hand in Zoom, or type down the questions in the chat.
Should I stop sharing?
No, it's fine.
Yeah.
You can
Okay.
you can keep it.
Yeah.
I don't see any raised hands right now. So let me start with a question. Have you ever thought about testing if you can modify it on your own. Like for example, if you have memory-like cells, T cells, what would happen if you start damaging their mitochondria? Would they become more short-lived, like effector cells, and vice versa? Could you actually increase the lifespan of the effector T cells by increasing their autophagy or improving their mitochondria?
Yeah, yeah, that's a good point. You know, I think to have a more definitive or causal link between the inheritance of mitochondria in different flavors, like damaged or not, and the fate of the cells. I think I haven't done it yet. So one of the things we would like to do is actually to, and this is something that we already have started doing in the lab, not looking at old mitochondria necessarily, but with Anna, that's in Barcelona, is to look at mitochondrial transfer. So basically, is a cell that has just healthy mitochondria, if we would push like some old mitochondria inside, would they then become more effector-like, or would they very, very quickly be able to get rid of this old mitochondria and they would stay the way they are? And the opposite, are cells that just have damaged mitochondria, for example, cells from aged animals, they really accumulate aged mitochondria. If we would give them young mitochondria, would this be enough to rescue their phenotype? Of course, we could do it in a more simple way, like more bulk way, by just like inducing autophagy, for example, in the cells, which can be done like, it doesn't work so well with mTOR inhibition in T cells, unfortunately, but could be done, for example, with spermidine. And then this would boost autophagy in the cell, but this would also do other things, not just increase mitophagy, and then this causal link would be a bit more loose, right? But yeah, this is something that I would like to explore, basically how to modulate this mitochondrial inheritance or mitochondrial health. And then one thing, I see that there are some other questions, and I'm going to try to just like finish this answer, but one thing that oh, now I kind of lost the so we are talking about basically rescuing mitochondria. Yeah, I'm gonna think about it later. Like I wanted to finish with some other idea, but now I lost track. I will come back to it probably.
Actually, that was my original question. If you could push the transfer of mitochondria in one way or the other, but I wasn't sure if it's even possible. Okay,
Yes,
so I can see that.
yeah.
Yeah, so it would be really cool to see your results on that. So I see that Anna has raised the question for their hand first. So please ask.
So I think this is really fascinating, and the whole thing with asymmetric cell division, and you pointing out that organelles of different age go to different daughter cells. But when you did this, why do the autophagy, or why do the mitochondria are kept healthy in the proximal daughter cell? Is that so, and not in the other? Is it also asymmetric division of autophagy components?
Yeah. So basically, what we see when we look at so there are ways to image autophagosomes. One of them is to look at LC3B staining, because LC3B is something that decorates, I think this was in one of my slides, it decorates the autophagosome. And LC3B is basically becomes lipidated and membrane bound when it's in the autophagosome. So basically, if you look at clumps of LC3B, this is a way to basically see where the autophagosomes are. And when I came to the lab, I thought, like, I already had the hypothesis that of course the memory-like daughter would be the one doing more autophagy, and then this would be the one that would have more autophagosomes. But actually, it looks like the opposite. So basically, the effector-like cell, the proximal one, is the one that if you image for autophagosomes has more autophagosomes. And now our interpretation of that, because we also have done, of course, you cannot do this with knockouts, but if you get cells from aged animals, what happens is that you have a huge accumulation of autophagosomes. So what we think is that in the effector cell, you have more autophagosomes, but they are accumulating because you don't have active autophagy. And this would indeed suggest that the cell that is on the opposite side, the distal side, is the one that is basically turning over this autophagosomes faster, which is also then gonna, as a consequence, turn over mitochondria faster. But I think I haven't done experiments to clearly show that it's mitophagy that is happening more on one or the other cell. One thing that also suggests that it is the case, and it's not so, of course, straightforward, but if you would look at I'm just gonna go back to my slide. I don't know if you are seeing the slides too. But when we look at this first order cells here, I can use this. Basically, the old staining is something that happened days before, but the young staining is something that happened just like hours before. And the cell that it gets rid of the old mitochondria is also the cell that's getting rid of this very young mitochondria. And this cannot be done. The new mitochondria cannot be segregated away. This is too short. Like in 2 hours, the cells are not dividing to get rid of this young mitochondria. So the fact that the young mitochondria is being lost together with the old mitochondria also is, I think, strong evidence that you have more mitophagy going on in these cells here. But when we look at the autophagosomes, we actually see more autophagosomes on these cells here, which could be because they are accumulating as they are not fusing to lysosomes. And this could be because lysosomes in the effector cells are being engaged with mTOR activation. So I think we don't have enough cell biology knowledge about that, but I hypothesize that when basically these lysosomes are engaged with mTOR activation, they are not really fusing with autophagosomes, and that's why you have this accumulation there. I have actually some images showing this asymmetric distribution of LC3B in the bioarchive. In the bioarchive is already there, and then it shows that also in the old cells, it's super accumulated because there is no autophagy happening there. And in old cells, we know that this accumulation of autophagosomes happens because the lysosomes are not working very well. And then that's why you don't have fusion of the autophagosomes to lysosomes, and then they end up accumulating. So that's why we think it's happening something similar in the mitosnap high cells. I don't know if I answered your question on it. I hope yes. At least partially.
Okay, and we have...
Just before I move on, I remember what I wanted to say now before. One thing we wanted to do is to find biomarkers of organelle aging, because then we can do this work, not just in mirroring cells using this SnapTag mouse. We could then do this in human cells. And this is basically one of the first things I want to do in the lab, is to find the biomarkers. So what does an old mitochondria express that a young mitochondria doesn't express, and the same for peroxisomes. Yeah.
Yep, thank you. That's, again, sounds cool. So last question from Marta Engel.
Hi, thank you for a very fascinating presentation. I have just a question regarding valorization of this kind of research. So I'm a cancer researcher. So my question is, do you think, well, what I take from your work is that perhaps blocking autophagy might, according to your research, might enhance T cell stemness, right? That would enable so like T cells to engage new antigens in context of vaccination therapy, for example, or immunotherapy. Do you think that that's something that your research can be applied to in kind of cancer research?
That's what I write in the grants, but that's not what I don't know if I'm going to be the one doing it. But basically, the idea is that working with these cells, another advantage is, of course, that especially if we can show that this is also happening in human T cells, that if you manipulate T cells, you can perhaps, for example, we know that CAR T cells divide asymmetrically as well, for example. If we boost CAR T cell asymmetry or the asymmetric inheritance of mitochondrial CAR T cells, this might promote their stemness, yes. The thing is that if you block autophagy, just block autophagy chronically, like what happens is that the cells will become more effector-like and will die. And actually, they won't even become true if, like, depending on the time point, actually, if you block autophagy, this can also damage their proliferation, because autophagy is also important to provide building blocks to the cell. So I think it's hard to use inhibiting autophagy as a modulation strategy, and that's why we thought that maybe the opposite, like actually boosting autophagy, might be interesting, because in the cells that clear up mitochondria, you have this enhanced stemness. And then basically by boosting autophagy, you would have a higher proportion of cells that are stem-like and long-lived. And these cells might be interesting in the context of adoptive T-cell therapies, for example, because then maybe you would need less reinfusion, I think, events. But I don't know also how good it is to have, I think, for example, a CAR T cell there. You probably know better than I. If it's good to have basically these cells around for a very long time, and basically being reactivated all the time, if this would lead to some type of, I don't know, like immunopathology as well. But yeah, this is something that when I basically envision why I'm using T cells, the reason is because this can be translated. If we find ways to modulate the fate of the cells, this might be useful in different contexts to either halt the formation of effector cells and boost the formation of this long-lived memory cells that would then stay around. But now just like to finish, basically one thing that I'm really focusing on, or I would like to focus on in the next years, is on this possibility of transferring organelles between cells as a way to boost their function. Because we have observed, at least in murine cells, that if you get cells from an old mouse, and these T cells are not, they are not very good in adoptive transfers and re-challenge setups. If we co-culture them with young cells, and we see that there is mitochondrial transfer between from the young to the old, they become better. So this would be a way to basically make the T cells of someone that, for example, has lots of exhausted cells, or like someone that's already older, to be reinvigorated and then be used in T cell therapies. But it's a bit science fiction still. But we indeed see a lot of mitochondrial transfer, and sometimes it is unidirectional. So basically, if you have young and old cells, the young cell gets the mitochondria of the young, but the young doesn't really keep the mitochondria of the old. And it might be because of autophagy, of course, because the old cell is doing less autophagy. And then when it uptakes something that's exogenous, it stays there. And the young cell is still autophagy competent. And then when it uptakes something from another cell, it just gets rid of it. So it seems to be, basically, this mitochondrial transfer seems to be good, especially to reinvigorate cells that are dysfunctional.
And of course, you'd have to know which type of mitochondria, right? That would be more.
This we don't know. We have done just with bulk, like just looking at mitochondria in general, and this is one of the work packages basically of the grant, is basically which type of mitochondria is being transferred. It's possible that smaller mitochondria are easier to transfer as well, and these old mitochondria, for example, are very bulky. So I assume, just like hypothesizing now that this would not be the ones that are moving around, but the younger and healthier and smaller would be the ones that would be shared more easily. Yeah.
Wow, it's mind-blowing. In brain cancer, there's a lot of work coming up now showing that they're looking at how the transfer is happening, right? And you have these microtubules that go from one side, and you can actually even show it in vivo. So is that similar pathway you're looking at?
It's a mix. We didn't do the imaging showing the transfer.
Mm.
This is all unpublished. I'm just like blowing information here. But so I don't have the data to show you. But basically, what we used inhibitors of trogocytosis or this nanotubule formation. So we just used inhibitors of different pathways that could promote transfer of organelles. And it's a mix. There are lots of things that contribute. So when we did these different inhibitions, lots of more than one pathway contributed to the transfer of mitochondria. So it's not just nanotubules. And there is a good contribution of trogocytosis, just like basically, a bit more promiscuous and less targeted than nanotubules would be. Yeah, but these nanotubules are amazing. That you really see the mitochondria traveling inside the nanotubules in these papers. It's really cool.
Okay, thank you all for the attention and for listening to that very engaging talk and engaging discussion. So see you probably next year in Siena, and good luck with new lab.
Thank you so much for the invitation, and good to see you.
Thank you.
Bye bye.